Hello, good morning all of you. Today we'll be discussing the most probable question from chapter number 16. We are entering into the physiology part, human physiology. The first chapter is human physiology, digestion and absorption. So now we are going to the questions directly. Select the correct matching pair. Gastroesophageal sphincter. What is a sphincter? Sphincter is a circular muscle which is acting as a valve that regulates the flow of something. Okay. Gastroesophageal sphincter is regulating it is preventing the backflow of food from stomach back to the esophagus, and thus it is preventing the reaching of the food from stomach to the buccal cavity. This is gastroesophageal sphincter. So it is regulating the passage of bolus. What is bolus? Bolus is the spherical form of food that we make in our buccal cavity with the help of our tongue, mucus, etc. So that spherical structure of food is called bolus. Pyloric sphincter at the hind end of the stomach that regulates the flow of the semi-digested paste-like food called chyme. From stomach, it is entering into the duodenum. So it is regulating the passage of chyme. That's also correct. Ileocecal valve or ileocolonal ring. It is at the junction of the last part of the small intestine, ileum, with the first part, namely the cecum, first part of the large intestine. So ileocecal sphincter that will prevent the backflow of the fecal material from large intestine. Okay, so ileocecal valve prevent the flow of backward flow of this is prevented by this sphincter. Okay, so these are all sphincters that act as valves. Okay, regulating, controlling the flow of food from one part to another part. Final step in digestion. See, digestion takes place step by step in buccal cavity, in stomach, in intestine, etc. And the last digestion takes place in the small intestine. That is mainly in jejunum and ileum. Mainly in ileum. That last digestion takes place very close to the epithelium of the ileum, so that the end product can be directly absorbed. So it is very close to the mucous epithelium of the intestine, small intestine. Very close to the mucosa layer. Part of alimentary canal corresponding to functions listed below. See, absorption of some water, minerals and certain drugs. We are having a chart. We'll be having that chart here. Drugs coming in contact with the mucus. So we are having some drugs. In stomach, we are having water, sugar and alcohol. Absorbed, main nutrients are all absorbed in the small intestine because digestion is completed here, and the final products of digestion such as glucose, fructose, fatty acids, glycerol, amino acids are all absorbed here. In large intestine, we are having absorption taking place for water, minerals and drugs. Okay, so these are the different parts of the alimentary canal through which the digested food materials are all absorbed. Okay, now you can answer this question. Absorption of some water, minerals and certain drugs. Okay, so in stomach, what are all absorbed? It is not like that. It is secretion of mucus which help in othering, attachment of undigestible materials. So undigestible materials are all pushed to the colon, and the colon wall is having abundance of mucus secreting cells. They will be helping in othering of the undigestible material and converting them into a semi-solid paste. That is the function of colon. So water, minerals and certain drugs, it is colon. Secretion of mucus which help in also colon. Presence of submucosal gland. Only region and only gland in the submucosa is duodenum, and the gland is Brunner's gland. Submucosal gland is the Brunner's gland. So Brunner's gland is in duodenum. So duodenum is the second one. Okay. And pepsinogen, pepsin is an active form. It is produced in an inactive form, pepsinogen, by the gastric gland. So it is in stomach. Hydrochloric acid, 1. So 1 should be 4, 2 should be 3, and 1 and 2 should be for 3 colon. So that option goes to the 3. Regarding human tongue, freely movable, correct? The correct statement. It is attached on the floor of the buccal cavity by a fold of frenulum. It is attached by posterior and anterior end is free. Esophagus open to the cardiac part of the stomach. The first part is the cardiac one. See, this part is the fundus. The raised part is the fundus. This is the cardiac, and this is the pyloric. The esophagus is opening to the stomach at the cardiac. Correct. Okay. Glossopharyngeal nerve control the movement of the tongue. It is the 9th one. We are having the 10th vagus supplying different parts of the alimentary canal, and this mainly is a mixed nerve which is controlling the movement of the tongue. Okay. We also have the 11th cranial nerve, spinal accessory, which is exclusively for the movement of the, it is a motor for the movement of the tongue. All papillae in the tongue bear taste buds. Now, we are having 4 types of papillae. They are circumvallate, here at the back of the tongue, almost like an inverted V, we are having circumvallate. You have very few foliate type on the edges, red flat structures, large number of mushroom-shaped cushion-like structures called fungiform, and the most abundant is the filiform, filamentous structures, which are all most abundant. And this filament or filiform are having no taste buds on them. So all papillae in the tongue bear taste bud. Filiform have no taste buds. Okay. These papillae are all sensory in function. They are all sensing. They are sensitive to touch, pressure, etc. Some of them will be having taste buds also. But filiform is having no taste buds. Temporary milk or deciduous, it is also called deciduous. Why it is called deciduous? Deciduous means this will be falling off. Okay. So, the teeth that appear first, then they are all replaced by a second set. That is why it is called deciduous. Are replaced by a set of permanent or adult dentition teeth. Okay. The set of teeth found only in permanent teeth but absent in milk. See, this is the milk dentition, permanent dentition. There is no difference in the incisors, canines, but premolars, they make their appearance only in the permanent dentition. Similarly, the last one of the molar, it is called wisdom teeth. They make their appearance only once. So, premolars and wisdom teeth, the last set of molar, are monophyodont. They appear only once, only along with the permanent dentition. So, how much? This is 4 plus 4, 8. This one 4. That is 1 in each half of each jaw. So, 8 premolars and 4 molars are making their appearance. This is the full dentition in a permanent one. We are having 2 incisors, 1 canine, 2 premolars and 3 molars. Okay. Match the following. What are glissons capsules? Glissons capsules are the connective tissue covering around a lobule of a liver. We know that liver is having 2 lobes. Each lobe is made up of a large number of small units called hepatic lobules. This is the structure of a hepatic lobule. You are having a hepatic sinus in the middle, around which the hepatic cells, liver cells, are arranged in the form of cords called hepatic cords, between which you have blood-filled sinuses. So, glissons capsule is connected with connective tissue sheath of the lobule. So, one is Q. So, immediately you eliminated all of this. You eliminated this one, you eliminated this one, and you are having only 2 questions, 2 options, B and D. So, concentrate on these 2. Okay. Then, what is gallbladder? Gallbladder is a cyst which can store and concentrate bile. Okay, that is the function, gallbladder. Concentrate. So, it is 2 yes. Now, we eliminated this one also. Now, we came to the answer immediately. Okay, see, this will take more than 1 minute for answering this question, but you will be saving at least 40 seconds because of this technique. How? See, this is elimination technique. We have eliminated that. This is not the answer possible. Then, by the second option itself, we came to the answer, this one. Hepatic lobules. See, what are all hepatic lobules? These are all the structural units of liver, structural and functional unit of the liver. Right lobe of the liver is largest. Actually, there are 4 lobes. In our book, it is given only 2, which are the major, a right lobe and a left lobe. Below the left lobe, below the right lobe, you are having the gallbladder. So, what is the right lobe? Right lobe is the largest part of liver. That's why we use the term largest. Okay. If you are considering only 2, larger. The right one is larger. Okay. Largest. Kupffer cells, macrophagic cells, which are all associated with the hepatic cords, are called macrophagic cells, are called Kupffer cells. So, phagocytic cells. Hepatocytes, that is the cells that are going to the composition of the liver, that is responsible for the production of the bile. So, 6 should be produces bile. And cystic duct, the duct of the gallbladder, is called cystic duct. We'll be again coming to the cystic duct in one of the later questions. Select the true statement. Absorption happens in mouth, correct? Stomach, small intestine, large intestine. Correct statement. Chymotrypsinogen. What is chymotrypsinogen? It is an enzyme produced by the pancreas. Okay. This is a gen. See, protein digesting enzymes are produced in an inactive form called chymogen. They are to be activated. That's a mechanism for protecting the gland itself from the action of the enzyme it produces. Pepsinogen, trypsinogen, chymotrypsinogen, procarboxy. It is a pro. Pro means inactive. So, we use the term either pro or gen to denote that it is an inactive enzyme. An inactive enzyme is also called chymogen. So, chymotrypsinogen is activated by trypsin, correct? Trypsin is having 2 activities. One, it can act on proteins. Two, it can activate other protein digesting enzymes of the pancreas like chymotrypsinogen and procarboxypeptidase. Enterokinase does not involve in breaking the peptide bond of macron. See, enterokinase is an enzyme present in the succus entericus. This will be only an activating enzyme that will activate trypsinogen. Trypsinogen is an important enzyme of pancreatic secretion. Trypsinogen is converted into active trypsin. See, trypsinogen is a gen, it's an inactive proenzyme, and this is converted to trypsin with the help of the enzyme enterokinase. Enterokinase is present on the succus entericus, intestinal juice. This is only an activating enzyme. It will activate trypsinogen to trypsin. Now, the trypsin will activate other enzymes. What are they? As I told you, procarboxypeptidase into carboxypeptidase and chymotrypsinogen into chymotrypsin. Okay. Enterokinase is only an activating enzyme. It will not be acting on any food ingredient. So that statement is a correct one. Renin, spelling is N, double N. That is the enzyme present in the children in small, which is responsible for the curding of the milk. It is absent in adult gastric juice. Correct. It will be gradually decreasing in amount as the baby grows and will become completely lost. Okay, so renin is absent in adult gastric juice. So all are correct. Enterokinase does not involve the breaking of peptide bond of macronutrients directly. It is only an activating enzyme. That's why it is also correct. Now, what is bolus? Yes, that was what I was telling you. See, once you take the food to the buccal cavity, we roll it with our cheek, tongue, etc. and convert it into a mass of ball-like structure. That ball-like mass of food is called bolus. That's how we push it to the pharynx and to the gullet. It is pushed to the esophagus, conducted by the peristaltic movement of the esophagus to the stomach. That is called deglutition, swallowing. So, how the bolus is formed? It is formed by the sticking of the food particles with the help of saliva. Saliva is containing mucus. Okay, so mucus. So one is C. So immediately we eliminated all other except D and A. Now you concentrate only on this one. 2 is same. Okay. What is chyme? Chyme is the semi-side digested food that is coming from stomach. So chyme is churning movement of the stomach, semi-digested. Both physical and chemical digestion is taking place inside the stomach. That physical process of rolling and pulverizing of the food within the stomach wall or within the stomach by the muscles of the stomach wall is called churning movement. Okay. What is chyme? The food after coming out of the stomach mixes with the bile, and in the intestine it is a fluid. Okay. It mixes with the secretions like bile, pancreatic juice, etc. and forms a fluid. Chyme is a semi-solid, whereas chyle is a fluid. So we simply say that chyme plus bile is chyle. So for easy remembering, chyme plus bile is chyle. So chyle is where we'll be having reaching the duodenum. Okay. So chyme will be formed as a result of churning movement. Chyle, that is coming from stomach, mixes with the secretions of the pancreas as well as bile that forms the liquid part in the duodenum. Then what is mastication? Mastication is simply chewing with the help of our teeth. So we are having dentition, A. Okay, we are masticating. Masticating means chewing with the help of our teeth. Okay, then we have peristalsis. What is peristalsis? The movement of the conduction of the food through the esophagus. So it is esophagus. And deglutition. What is deglutition? It is swallowing. Simply, we can say that it is swallowing. So it is with the help of the glossopharyngeal nerve. In one of the earlier questions, there was glossopharyngeal nerve controls the movement of the tongue by which we are all pushing. Swallowing is with the help of it is the ninth cranial nerve. Clear? Swallowing is with the help of that ninth glossopharyngeal, ninth cranial nerve. So we came to the answer all correctly. Match the following. Sodium taurocholate, sodium glycocholate and taurocholate are the two types of sodium salts produced by reacting with the products of the liver. They are all produced by the liver, not waste material as bile pigments. That's an important point. That's an important difference between the bile salts and bile pigments. Bile pigments are all waste materials, whereas bile salts are all produced for a purpose. Sodium taurocholate and glycocholate. Okay, so they are all helping in emulsification of fat. This is a bile salt. Bile salts are emulsifying the fat and also used for making micellus, small droplet after the digestion. Salivary amylase, it is called thiolin. It causes partial digestion of starch. We know that the starch will be 30% digested by the salivary amylase. Pancreatic lipids, that is the most powerful lipid digesting. It is called stepsin. That word is not in our books. Stepsin. So, converting triglyceride substrate found in ingested oils to monoglycerides and free fatty acids. So, fatty acids and glycerols are converted. Okay, so that is the very powerful pancreatic lipase. Then trypsin. Trypsin, pepsin are protein digesting. So what they are all doing, they are all responsible for the breaking down of protein, proteolytic enzyme present in pancreas. Pepsin is present in gastric juice. Trypsin is. What is the main difference? Pepsin is in acidic medium, whereas trypsin is in highly alkaline medium, acting. Then renin, spelling is double N-I-N. You should be very careful to the spelling. R-E-N-I-N is a hormone produced by the kidney. Okay, which is playing an important role in regulation of GFR. This is renin, that is digestion of protein of the milk, casein. Okay, so the option goes to apple. And for the true statement, small amount of monosaccharide like glucose, amino acids and some electrolyte like chloride ions are generally absorbed by simple diffusion. Absorption takes place by different mechanisms, and this is one of the simple mechanism. Without energy, by diffusion, they are all absorbed. Small amounts. That is why it is given small amount, not complete. Complete absorption takes place by active digestion, active absorption with the help of energy. Here it is small amount like glucose, amino acids, etc. are all absorbed. Chylomicrons are transported into lymph vessels in the villi. What are chylomicrons? After absorption, the fatty acid and glycerol are rejoining to form large molecules of triglycerides. These triglycerides are then covered with a coating of protein, beta protein. Thus, it becomes miscible with water. Such large lipid molecules with a protein coating is synthesized within the Golgi complex of the epithelial cells. They are called chylomicrons. So, there are chylomicrons transported into the lymph. That's how they are all coming out of the epithelial cells. Now they are all taken up by the villi, okay, and transported. Absorption of water, simple sugar and alcohol takes place in stomach. They will be absorbed in the stomach. Simple sugar, water and alcohol are absorbed in stomach. Because alcohol requires no digestion, it can be absorbed in the stomach itself. So, only two are correct, the third one is wrong. So, the option goes to cat. Now, we are having another set of questions where we have to select the word, appropriate word. The tongue is attached to the floor of the oral cavity, that is buccal cavity, by a ligament called or ligamentous fold called frenulum. So it should be frenulum. So whenever such a multiple statement, fill in the blank question is given, you read the statement and find the answer. What should be the correct option? Upper surface of the tongue has small projections called papillae. We have discussed it, papillae, which are all sensitive. The long coiled mid portion of the small intestine. It is long coiled, but mid portion. The small intestine is having duodenum, which is uncoiled. Jejunum, elongated, slightly coiled. The maximum coiled is ileum. So, duodenum, jejunum and ileum. These are the three in that order. So, what is the middle one? Jejunum. Okay. Now, you selected the word frenulum, papillae, jejunum. Now, what is that order? What is that option that is having these three in the order, correct order? It is C. So, that is the answer. Match the following are one of the most important type of questions in all NEET examinations in biology. Curding of milk. Okay. It is a bit confusing, but also helping you if you understand the technique of answering the question. Curding of milk is with the help of renin. So, one should be A. You immediately eliminated other options. You concentrate only on these. One is A, then 2 is B here, 2 is D. So, that will be deciding factor. Emulsification. What is emulsification? We are all breaking lipids into tiny droplets. That is called emulsification. For that, we will be having the bile salts helping. The fat is broken down into tiny droplets. Okay. Now, these tiny droplets are formed by the help of bile salts. So, emulsification, bile salts. 2 should be dal. Here it is B. It is not fructose. Now, immediately you came to the answer. Though it's a lengthy one, you can come to the answer very freely, very quickly if you know this technique of answering. Okay, then anti-microbial agent, which is antimicrobial, which is against the microbes. The lysozymes are antibacterial. Bacteria are all unicellular. Okay, so microbes. So antimicrobial, antibacterial, lysozyme, enzymes. Facilitated transport. What is facilitated transport? It is a passive transportation where the transportation is facilitated by, assisted by a carrier protein. Fructose is transported, so it is for fructose. That's the clue. Okay. Facilitated transport. 4 is B. See, if you know that facilitated transportation is for fructose, if you are all confused with other options, no problem. Facilitated transport 4 is B. You go for that. Only one is having, only this one is having that option. So immediately, even if you are not aware of other options, one pair will be helping you in identifying the correct option sometimes, very often. So you look for that, so that you will be immediately coming to and you can confirm that this is the right answer. Okay, and absorption of fat. How fat is absorbed in the form of micellus. What is micellus? After emulsification, the tiny droplets of fatty acids are all given a coating. They are all covered with a coating of bile salts. It is with the help of the bile salts, the fatty acids are all broken down into tiny droplets and thus it forms a coating around them. That is what is called micellus. Micellus are very small. So, what is micellus? Fatty acid plus bile salts. They will be forming micellus. In the form of micellus, they are all absorbed into the epithelial cells and within the Golgi complex, enzymatically, the fatty acids are converted into triglycerides, large molecule triglycerides and protein coat. That is given called as chylomicron. Chylomicrons are larger ones, almost having the size of 1 mu. Okay, so fatty acids first convert into micellus, absorb, then again resynthesized to triglycerides. They are all given a coating of protein, thus they are all converted to chylomicron and then released from the epithelial cells, reaching the lacteals. That's how they are all absorbed. Okay, so the answer is very simple if you know the technique. Select the incorrect match. See, here I have given the diagram of the ducts of pancreas as well as the liver. What are Peyer's patches? Peyer's patches are some masses of lymphoid tissues in the submucosa region of the small intestine. Submucosa region of the small intestine. So, submucosa. They are lymphoid tissues. Masses of lymphoid tissues. They form malt, a major part of mucus associated lymphoid tissue. Okay, malt. Ampulle of Vater. See, these terms are not in our NCERT books. What is ampulle of Vater? The hepatopancreatic duct, the common hepatopancreatic duct, is expanding to form an ampulla. That is called ampulle of Vater and is opening to the duodenum, which is controlled by the sphincter of Oddi. So, ampulle of Vater, hepatopancreatic duct, correct. Hemorrhoids. What are all hemorrhoids? The veins of the rectum will be swelling and blood become accumulated, which is commonly called as the piles. That may be commonly called as the piles, hemorrhoids, rectal veins. It is because of the swelling. The walls become very thin, they become bulged out and you will form the hemorrhoids in the rectum. That is forming or that may be leading to bleeding. That is called piles. Thoracic duct. What are thoracic duct? The lymphoid ducts are all joining to form two major lymphatic ducts. One is the thoracic duct on the left hand side, which open to the subclavian vein coming from the left arm. The other one is right thoracic duct on the right side here. That will be opening the vein coming from the right arm. That's how the lymphatic fluid is, the lymph is again joining the blood. So remember, small lymph vessels are all joining to form large ducts. And along with that, you will be having lymph nodules or lymph glands. Now, these ducts are all joining to form two larger ones. The most important one is the thoracic duct, which is the lymphatic duct, which is present on the left hand side, on the left abdomen. It joins to the left subclavian vein, and the other one is present on the right hand side, right thoracic duct. So on the left side, you are having the thoracic duct and right lymphatic duct on the right side. These are the two important lymphatic ducts by which the lymph is pouring back into the blood. Tinea coli. What are Tinea coli? In the colon, remember, the colon is having sac-like structures called hostrae, which is helping in the forward pushing of the undigestible materials. And on the surface, you have three elastic bands on the surface. These elastic bands are called Tinea coli. These elastic bands are helping in recoiling, that is coming back of the hostrae. See, hostrae will be pushing. When they push one by one successively, the inner content, that is the fecal material, is pushed to the next chamber, and that's how they are all moving. The fecal material are brought to the rectum finally, but the hostrae has to come back. This coming back, recoiling, is with the help of the elastic bands, three elastic bands present on the surface of the colon. That's why it is called Tinea coli. So it is not in duodenum, it is in colon. Is that clear? This is the position of the sphincter of Boyden and sphincter of Oddi. Yes. Which of the following is required for the emulsification of fat? Emulsification is breaking down of the fat into tiny droplets, that is assisted by sodium taurocholate and sodium glycocholate, which are commonly called as bile salts. Bile salts are all produced by the liver. It is the secretory product. Don't confuse with bile pigments. Many children are having confusion between bile salts and bile pigments. Bile pigments are the excretory waste products formed from the broken down RBC hemoglobin. So that hemoglobin is converted into their colored products, waste products, bilirubin and biliverdin. Verdin is green in color, biliverdin. Bilirubin, yellowish brown in color. Okay, so it is not HCl, bile pigment, but bile salts. Pepsin differs from trypsin. Both of them are digesting protein, acting on protein, proteolytic. Both of them are produced in inactive form. Pepsin is produced in the gastric glands. It is acting in acidic condition. Its pH is, optimum pH is 1.8. Okay. Whereas trypsin is in pancreatic secretion. Pancreatic juice require an alkaline secretion 7.8. So pepsin differ from trypsin in that they digest in alkaline medium in the stomach, no. Protein in the alkaline medium of the intestine, no. Protein in the acidic medium of the stomach. Pepsin is in the protein in the acidic medium, 1.8 in the stomach. Yes. Which of the following statement regarding bile is true? Okay. Bile is a watery greenish fluid, correct? Containing bile pigments, bile salts, cholesterol, phospholipids, etc. Correct? It does not contain any digestive enzyme. Bile pigments play an important role in digestion. Bile pigments, see, bile pigments are not playing any role. It is bile salts. bile salts, sodium taurocholate and glycocolate. Bile salts emulsify correct fat particles, thereby fat droplets acquire larger surface area. That's the main function. Correct. Enzyme lipase, a component of bile. No, bile is not containing any digestive enzyme, but it is highly essential in digestion of the fat. How? In three ways. One, it emulsifies fat, break down into small droplets so that the surface area is increased. Two, it activates the lipase mainly present in pancreas and succus entericus secretions. Three, it will be also helping in absorbing, bile salts are helping in absorbing the digested fatty acids as micelles. So these are the three ways in which the bile is helping in fat digestion. So it is having no digestive enzyme. That is wrong. So regarding bile, only two are correct. 1 and 3 are correct. These two are all wrong. So we go to the option. Which of the following is the correct matching site of the action of the given substrate, the enzyme acting upon it, and the end product? Small intestine, protein, pepsin. Pepsin is present in stomach, not in intestine. Okay, that's why it is wrong. Stomach, fat. Actually, fat is not at all digested. The gastric juice contains very weak gastric lipase, which is not at all responsible for digestion. So, micelles is never produced. Micelles is produced only in the small intestine, not in stomach. Small intestine, starch, 70% of the starch is digested with the help of the amylase present in pancreas, correct? And alpha amylase, both salivary amylase and pancreatic amylase in human beings are alpha amylase because the the glucose produced is alpha 1. It is converted into the disaccharide maltose, correct? That is correct one. In duodenum, triglycerides, trypsin. See, triglyceride is a fat, trypsin is acting on protein, so it is not at all correct. Buccal cavity, starch, tyalin. Tyalin is acting on starch, that part is correct. 30% of the starch is converted into not glucose, it is converted into maltose. Maltose is a disaccharide, glucose is a monosaccharide. Only at the end of digestion, glucose, fructose, etc. are all produced. That is only in the small intestine. Complete and final digestion takes place in the small intestine, not in buccal cavity. It is disaccharide. Okay, maltose is a disaccharide. It is to be acted upon by maltase that is present in the succus entericus. Goblet cells. What are goblet cells? Goblet cells are mucus producing. In one of the earlier chapters, I have told you that goblet cells are typical example for holocrine. Complete cell is burst and secretions are coming out. They are present throughout the alimentary canal. What are they? They are all modified epithelial cells. What type of epithelium alimentary canal is having? They are lined by tall columnar epithelium. So there should be columnar epithelial cells. Chondrocytes are cartilage cells, compound epithelial cells, not at all lining the alimentary canal. Squamous epithelial cells. No, it is not squamous epithelial cells. It is tall columnar cells that line the alimentary canal. Now, this is regarding the histology of the alimentary canal. What are the 4 types of layers? Identify the correct statement with the human digestive system. Serosa. The outermost one is the connective tissue, thin connective tissue layer, which is the serosa. Serosa is the innermost. No, it is the outermost. Ileum is a highly coiled part. That is the most coiled part. Is that correct statement? Vermiform appendix arises from the duodenum. No. Vermiform appendix arises from the cecum. Cecum is very small in our case, whereas it is very large in herbivores. Okay. It harbors symbiotic bacteria. Okay. Which are all very, very helpful for us. So here, vermiform appendix is not arising from the duodenum. It is arising from cecum. Ileum open to the small intestine. No. Ileum open to the cecum. Ileum is the last part of the small intestine. It open to the first part of the large intestine, namely the cecum. Cecum, colon and rectum are the three parts of the large intestine. So that also is wrong. So the only correct one is 2. Enzyme enterokinase help in conversion of. Remember, enterokinase is responsible for the conversion of, it is an activating enzyme which is responsible for the conversion of trypsinogen to trypsin, not pepsinogen to pepsin. It is marked wrong. Enterokinase, it is an activating enzyme present in Succus entericus. It activates trypsinogen to trypsin. Okay. Caseinogen to casein. No. It is in milk, stomach. Protein to polypeptides. No. Trypsinogen is converted into active trypsin. In one of the earlier questions, we have discussed it. Okay. Hope you will be remembering it. Again, the primary dentition in human. What is the main difference? In milk dentition, there is no representative of premolars. Premolars will appear only if the permanent dentition. And the last molar, that is the wisdom teeth, also make their appearance only very late. So we are having incisors, canines and molars, but no premolars in milk dentition. 0/0 dental formula. Enzyme that is not present in Succus entericus. What is Succus entericus? Simply called as the intestinal juice. It is produced by the crypts of Lieberquin, which are all lying in between the adjacent villi. They are having a large number of enzymes which are responsible for the complete and final digestion of all types of food ingredients. Lipases. Lipases are present in the pancreas also, in stomach secretions, gastric juice also, lipases are present. Okay. Maltase. It is acting on maltose, breaking the maltose into glucose. Correct. So enzyme is present in Succus entericus. Nucleases. Nuclease is the enzyme that is acting on nucleic acid. It is present only in pancreatic secretion, pancreatic juice. See, nucleic acids are first converted into nucleotides with the help of the nucleases that is present in pancreas. Now, these nucleotides are converted into nucleosides by breaking away the phosphate group or phosphoric acid with the help of the enzyme nucleotidases. Now, the nucleosides are acted upon by nucleosidases, so that nucleosides are converted into sugar and phosphate. These two are all present in Succus entericus. Nucleases is present in pancreas. So, enzyme that is present in Succus entericus include lipases, maltases, nucleosidases, but not nucleases. So, nucleases is present only in pancreas, pancreatic secretions. Okay. So, we take 3 as the correct answer. Which hormone do stimulate the production of pancreatic juice and bicarbonates? See, the alimentary function are controlled by both nervous system and endocrine system. We are having a number of local hormones which are all locally controlling the secretion of the enzymes of the glands, functioning activities of the alimentary canal. For example, we are having gastrin secreting. The first hormone that is discovered, that is secreting itself, is produced by the duodenal part, duodenum. That secreting production of pancreatic juice, it is having two actions. First, secretin suppresses the gastric glands. Two, it activates the first secretion of the pancreas. First secretion of the pancreas is the alkaline secretion, watery secretion which contain water and bicarbonate only. That is the function of secreting. Then second one is pancreozymin cholecystokinin, CCKPZ. CCKPZ, they are all produced by the special cells of the duodenum. CCK act on cholecysto, bladder of gallbladder. Okay, they act on gallbladder so that gallbladder contracts, emptying the bladder. Bile is coming out. Okay, so CCK, cholecystokinin, PZ. This PZ, pancreozymin, act on pancreas, causing the second secretion of the pancreas to come out. That will contain the enzyme part. Okay, so production of pancreatic juice and bicarbonate, that is the clue word. Bicarbonate, first secretion. Second secretion contain enzyme part of the pancreas. Okay, that is under the control of pancreozymin, cholecystokinin. So production of pancreatic juice and bicarbonate is under the control of CCK, cholecystokinin, and secretin is responsible for the bicarbonate. Clear? Cholecystokinin is responsible for the contraction of the gallbladder. Then crypts of Lieberkühn secrete what? Antibacterial lysozyme. Which cell? We are having mainly three types of cells in the intestinal glands or crypts of Lieberkühn. What are they? They are all goblet cells that produce mucus, Paneth cells that produce all enzyme part, and enterocytes. Enterocytes are producing bicarbonate and watery secretion. Okay, so here the question is antibacterial lysozyme, which is an enzyme. All enzymes are produced by the Paneth cells. What are all argentaphin cells? Argentaphin cells are cells which are responsible for the production of local hormones. Chymogen, inactive. Pancreas is having no chymogen. Okay, it is producing a protein digesting, which is that dipeptide or erupsin. It is called erupsin. It is not at all producing any chymogen. Chymogen is an inactive protein digesting enzyme. So Paneth cells, correct. Kupffer cells. What are Kupffer cells? Kupffer cells are large macrophagic cells which are all responsible for breaking down of pathogens, dead cells, etc. They are macrophagic cells which are all present in liver. Kupffer cells. Clear? So it is not present in crypts of Lieberkühn. Crypts of Lieberkühn are the intestinal glands. Hepatic portal vein. What is a portal vein? A portal vein begins in small capillaries and end in capillaries. They carry something. Okay, from small intestine, all the digested materials are absorbed and carried to the liver. They end up in liver. That is why it is called portal. Hepatic portal. Hepatic means which is carrying material to the liver. Hepar means liver. That's why we use hepatic artery, hepatic veins, okay, hepatocytes, etc. So hepatic portal veins drain blood to liver from small intestine, stomach, etc. That's how absorbed materials are all carried to the liver. Then a baby boy age 2 years is admitted to the play school. He is only a small baby. So this is just to confuse you, giving you a lot of words. But the question is, he is having only milk dentition. Okay. You have to think and understand that it is a clue for that milk dentition and passes through the dentist checkup. The dentist observes that the boy has 12 teeth, only 20 teeth. That is milk dentition. Which teeth are absent? In milk dentition, what is the formula? Premolars are all missing. We are having incisors, canines. Premolars are missing. Only 2 molar each. So, there is no representation of the premolar. That is what is meant. Okay, actually, then, which of the following option best represents the enzyme composed of pancreatic juice? Remember, pancreas produces 2 types of secretions. The first secretion is only an alkaline. It contains water bicarbonate. Okay. This is under the control of secretin. The second secretion, pancreozymin. It is enzyme containing all the enzymes. What are the enzymes? Amylase, peptidase, trypsinogen, renin. No, renin is not present. It is produced by the gastric glands. So here also you are having renin. Okay. Amylase, pepsin, maltase. It is present only in succus entericus. So we have eliminated the first 3. Lipase, the most powerful enzyme which can digest lipid, are present in the pancreatic juice. It is called stepsin. Amylase, 70% of the digest of the starch. Amylase, pancreatic amylase. Okay. Trypsinogen, inactive form, procarbopeptidases. These are all protein digesting enzymes of the pancreas. So that is the correct answer. Which of the following gastric cells indirectly help in erythropoiesis? What is erythropoiesis? Conversion and production of mature RBC. Formation of mature RBC can be simply called as erythropoiesis. Formation and maturation of RBC. They are all under the control of vitamin B12, cyanocobalamin. That B12 is Cassell's factor. What type can it is? Cassell's extrinsic factor. Intrinsic factor is that is present inside. Cassell's intrinsic factor is helping in the absorption of that vitamin B12. Vitamin B12 is the extrinsic factor. Cassell's extrinsic factor that we get sometimes from the food, and this is absorbed and utilized for the maturation of RBC. For the maturation of RBC, we need cyanocobalamin, vitamin B12, without which no RBC can mature. RBC maturation. Otherwise, you will be having immature large nucleated biconvex cell. This biconvex cell is to be converted into plate-like enucleated mature cell. Then only they can carry oxygen. This is the RBC, mature RBC. This is the erythroblast cells. This is erythrocyte. This conversion is called maturation. For this maturation, we need vitamin B12. And B12 is absorbed with the help of Cassell's intrinsic factor, which is produced by the parietal cells or oxyntic cells. Oxyntic cells or parietal cells are producing HCl and Cassell's intrinsic factor. That Cassell's intrinsic factor is responsible for absorption of vitamin B12. Only with the help of the vitamin B12, the erythroblast will be converted into erythrocytes. Now you got it? Without which you will be having pernicious anemia, that condition where the blood cells are having more erythroblast and less amount of erythrocyte is called pernicious anemia. So a deficiency of cyanocobalamin will be causing pernicious anemia, and parietal cells are producing that Cassell's intrinsic factor that is responsible for absorption of B12 from food. That's why we say that the parietal cells are indirectly helping in erythropoiesis, formation of mature RBC. Which of the following terms describe human dentition? Pleurodont. What is pleurodont? Teeth are kept in pleura. Pleura is the membrane that cover the bone. No. We are thecodont. They are kept in sockets. Cavities called sockets. Correct. Diphyodont. 2 sets of teeth. Correct. Heterodont. Different type of teeth. We are having 4 types of teeth. Incisors, canines, premolar and molar. So that's the correct one. It is thecodont, diphyodont, but not homodont. Homodont means all teeth are same. That is what is seen in reptiles. All teeth are all same. It is pleurodont? No, it is not at all pleurodont. Okay, so only correct term is 2. And coming to the last question of this chapter, identify cell whose secretion protect the lining of gastrointestinal tract from various enzymes. See, even if you are producing HCl, that HCl is not coming in contact with the epithelium of the alimentary canal. It is protected from excoriating activity of the HCl. How? With the help of a thick layer of mucus. See, throughout the alimentary canal, respiratory tract, etc., we are having goblet cells that produce mucus. In alimentary canal, the mucus is having a number of important functions. One, it provides a slimy inner coating so that food can pass without injuring through the intestine smoothly. Second one, it will cause othering of the food particles together. It will also act as a physical barrier, protecting the lining of the alimentary canal. It will be physically protecting the alimentary canal from the activities of the enzymes. Bacteria cannot also enter because, remember, this is the epithelial cells. The epithelial cells are given a very thick coating of this. Okay, this is the mucus lining so that the pathogen bacteria cannot enter into the epithelial cells easily. It acts as a physical barrier. Clear? So, gastrointestinal tract, who is producing this mucus? Goblet producing unicellular cells, which are all modified epithelial cells of the alimentary canal. So, goblet cells. Oxalic cells are the cells present in the gastric gland that is responsible for the production of Cassell's intrinsic factor and HCl. Duodenal cells. Duodenal cells are only cells of the duodenum. Here it is throughout the alimentary canal, protecting the entire lining of the intestinal. Chief cells or zymogen cells are the cells of the gastric glands which are producing zymogens. So the answer goes to goblets. Clear? Okay. I hope now it is thorough to you. The chapter is thorough to you. Almost all concepts are touched. Almost all different types of questions are also included in this one. So understand that this is the type of question and the content which is likely to be asked this year also. And with that, I conclude this session and will come with another set of questions from respiration next day. So thank you all. Thank you very much. Okay. We will meet with the next set of questions next day. So thank you. Thank you very much.